Welcome to Ask the Expert. Today's guest is Slavica Tudzarova. She's coming to us from UCLA, from the Hillblum Islet Research Center, and the title of her talk is The B Cell or Beta Cell Uphill Battle in Type 1 Diabetes, Discovery of the Game Changer. A little bit about her is Dr. Tudzarova's research is focused on the crosstalk between the metabolism, DNA replication, initiation, and cell cycle in human cells and tissues. Defects in these processes commonly occur during malignant transformation or human diseases like diabetes and neurodegeneration, and they involve abrogation or checkpoint control and unscheduled replication, which leads to apoptosis and cell loss. As part of this program, she's investigating the abnormal mitochondrial morphology and function in diabetes that interferes with sustenance of B-cell replication during compensatory regeneration. Her group's now working on fostering B-cell regeneration through strategies that restore normal mitochondrial network and function, thereby preserving functional B cells. After obtaining her BS in pharmacology at University of St. Cyril, Macedonia, Dr. Tudzarova underwent training in tumor biology at University of Vienna, Austria, where she obtained her PhD at the University of Tumor Biology and Cancer Research. She then joined University College in London in the UK. She was a postdoctoral fellow and established her independent research program funded by Ernst Young SIFTA and Industrial Research Collaboration before appointed to assistant professor at Larry Hillblum Islet Research Center at the David Geffen School of Medicine at UCLA. Welcome, Dr. Tudzarova. Thank you so much for joining us. I'm really excited to hear more about your work and its very interesting premise.
Thank you so much, Monica. Thank you to you and the whole team. And I'm absolutely delighted to have this opportunity to share my work in front of sugar science community. I entitled my work as a beta cell uphill battle in type 1 diabetes, and it will come throughout my talk why I called it this way, the beta cell uphill battle, almost significant of the beta cell Sisyphean work or uphill rolling of stones as a challenge in type 1 diabetes. And then I will talk about discovery of the potential game changer. So long ago, it has been now established that type 1 diabetes is not only a T-cell mediated autoimmune disease. And there is accumulating evidence to support that. It's coming from a lack of durable effects of immunosuppression to contain the autoimmunity in type 1 diabetes, decoupling of islet autoimmunity from development of type 1 diabetes, for example, through low rate of insulitis at the time of diagnosis and onset of type 1 diabetes. But also strikingly puzzling is the unexpectedly high proportion of beta cells that persist, although they do not always function after diagnosis of type 1 diabetes. And now beta cells are moving to a new spotlight in investigating pathogenesis of type 1 diabetes from various reasons. So first of all, during the stress, and here on the right, you can see quite a complex cartoon depicting that stress, beta cells can process different neoantigens and thus which become relevant for peptide presentation of beta cells. And they come from various types of stresses, but some of them are really a shared commonality with type 2 diabetes, for example, are ER stress and unfolded protein stress. So therefore, it is maybe fair to say, given that evidence, that beta cell stresses that contribute to beta cell dysfunction also contribute to neoantigen generation and exacerbate at the same time autoimmune reaction in type 1 diabetes. Now, further being in the spotlight, beta cells have been highlighted as potentially perpetrators that can propagate the autoimmunity of type 1 diabetes. And we recently acquired knowledge about the role and relevance of senescent beta cells. So beta cells that undergo injury early during the development. And because of that, they attain a program, which is actually a normal program that prevents genomic instability because restricts, in other way, saying beta cells from replication if they have undergone genotoxic lesion. So, but here the team presented that these senescent cells actually can be perpetrators because they're responsible for secreting pro-inflammatory agencies that can propagate type 1 diabetes. So therefore, they are more and more shifted in the spotlight away from autoimmunity, but as direct perpetrators in the onset of autoimmunity in type 1 diabetes. Now, what is interesting, and we probably all of us know this progression through the stages of type 1 diabetes, is the fact that progression from the onset and detection of autoimmunity to loss of sufficient beta cells for the onset of type 1 diabetes is relatively slow in adults with recent onset of type 1 diabetes. Therefore, the question is, what is the mechanism of this slow progression? What is the mechanism of the sustenance of the injury in those beta cells from the onset of autoimmunity before sufficiently numbers of these beta cells are lost to lead to onset of hyperglycemia? So we have made one very interesting observation, and here it is presented. So the presented are immunostaining in the pancreata from brain dead donors, brain dead donors with type 1 diabetes, where we have found basically that there is a very high extent of protein, increase in protein expression in two key metabolic regulators called HIF-1 alpha and PFKB3. So HIF-1 alpha comes from hypoxia-inducible factor 1A alpha, and PFKB3 it is its transcriptional target. So we found that up to 2/3 of all beta cells, which here are revealed in green, based on using specific insulin antibodies to reveal their identity, and in white, based on glucagon antibodies. So up to 2/3 of all these beta cells in brain dead donors with type 1 diabetes and residual beta cell mass basically show immunopositivity for the downstream target of this pathway, PFKB3 or phosphofructokinase. Now, this referred very specifically to beta cells and not to alpha cells. But interesting, it was also a shared commonality with type 2 diabetes. Now, you can easily argue that this is maybe because this is a phenomenon which is a consequence of hyperglycemia. However, we were lucky to get hold of a pre-diabetic case, pre-type 1 diabetes, that Dr. Peter Butler have extensively described and reported about, and we found half of the extent of this PFKB3 immunopositivity to describe this pre-diabetes. Also, we have the same type of evidence in rodent pre-diabetic models of type 2 diabetes, indicating that this pathway actually qualifies merely for potentially an underlying mechanism of diabetes, of islet failure in diabetes, rather than consequence of hyperglycemia. Now, interesting is that almost at the same time came evidence, it was published from genome-wide association studies, that in reference to this gene PFKB3, there is a single nucleotide polymorphism that extracts the low C in this gene, PFKB3, with highest, strongest susceptibility significance. And guess what? This was found not only to account for a late onset of autoimmunity in diabetes in adults, but also not only in LADA, but also to be true across type 1 and type 2 diabetes, basically indicating that our observation is supported by this, or very well aligned with these GWAS studies. Now, when we identify this pathway to be highly relevant for beta cells in context of type 1 diabetes, we ask whether only HIF-1 alpha and PFKB3 are singled out as relevant representatives of the pathways, or basically they are tip of the peak of the iceberg, which is a part of a wider injury response program. To identify and substantiate that, we looked at available RNA-seq data from comparing the genomes between the transcriptomes between type 1 diabetic donors, so human islets from type 1 diabetes versus non-diabetics. And we found actually that more genes from the transcriptional program of HIF1 alpha were affected. Here they are listed, and there was a significant overrepresentation, which was uniform across the RNA-seq datasets. When reconstructed this, what it means is, and if you can see under A and B are represented the pathway where these two relevant metabolic proteins belong, that means there was upregulation of a process which is known as aerobic glycolysis and also known as oxygen-independent way of generation of energy. So under B on the right side, you can see that highlighted in red what has been changed, which indicates that there is a genetic remakeup based on HIF1 alpha transcriptional activity, which basically breaks down the sensing of glucose by replacing glucokinase with hexokinase, then enhances the volume or the pace of aerobic glycolysis, and at the same time steals pyruvate into production of lactate via upregulation of this gene LDHA, but at the same time suppresses utilization of pyruvate from mitochondria. So indicates that not this pathway is part of epistatic, or these proteins are part of epistatic pathway of regulation of aerobic glycolysis that is relevant for type 1 diabetes. Now, why we were so much intrigued is that HIF1 alpha and PFKB3 actually belong to so-called disallowed genes. So basically, they are imminent and important during development of beta cells, but then they are suppressed in order to allow unseized, uninterfered, highly specialized beta cell function. And from this diagram here, it should become clear why they're suppressed after development of beta cells in mature and highly specialized beta cells. The reason is that both of them mediate through aerobic glycolysis, actually redirection of glucose metabolism away from mitochondria, thus mediating breakage or dislinkage between glucose sensing and insulin secretion, and directly being responsible for the loss of glucose-sensitive insulin secretion. Now, in order now to understand, since even in prediabetes, there are complex triggers potentially that can lead to upregulation of HIF1 alpha and PFKB3, it is important to distill what is the trigger, what actually leads to their upregulation. For this reason, we have used human islets, which we have treated with cytokine mix in order to reflect and mimic the metabolic and pro-inflammatory stress close to one seen in type 1 diabetes. And then we measured the protein expression and transcriptional levels of these proteins, of the transcription levels of these genes. We found that not only HIF1 alpha and PFKB3 are affected positively with upregulation, but also another two which belong to transcriptional targets of HIF1 alpha, such as hexokinase and LDHA. And quantification of these measurements are presented here from B to E. On top of it, we've seen some trend in increase in secretion of lactate. And important to say, because it has been often argued that human islets deteriorate in the culture, that this is shown, right, the potential bias of deterioration in the culture is kind of present in the control. And what we show is fold change relative to the cytokine mix treatment or pro-inflammatory stress. That was, of course, interesting to observe that also on protein level, we see increased expression of these proteins and also distilled to pro-inflammatory stress in relation to the human islets. But now we were interested in the consequence of HIF1 alpha and PFKB3 targeting during pro-inflammatory stress. In other words, what is their role? To understand this, we reproduced these experiments, again, using human islets, which we treated with cytokine mix, this time in presence or absence of inhibitor, here translational inhibitor of HIF1 alpha, KC7F2, at growing concentrations, so 2 concentrations, 5 and 10 micromolar. And then also we have used a strategy of post-transcriptional silencing of PFKB3, almost to ask the same question, but now in relation to PFKB3. So these results here, this Western blot here, is showing accumulation of cell death in reference to translational inhibition of HIF-1 alpha, but not of suppression of PFKB3, indicating that HIF-1 alpha is basically the one which is sufficient and necessary to regulate the vulnerability of beta cells, in this case, human beta cells in context of inflammatory stress, but not PFKB3. Now, since we are doing parallel studies in type 2 diabetes in these models, I have to say this is another commonality, so shared commonality between these two models, because we recently published that also the roles of PFKB3 and HIF-1 alpha in context even of type 2 diabetogenic stress diverge. So they have divergent role, although they belong to same epistatic axis of regulation. Now, this is further quantified here. As you can see at the G and J, you can see clear no difference in the cell death when PFKB3 is suppressed, but only when HIF-1 alpha, which means that for sustenance of these injuries through pro-inflammatory stress in beta cells, it is important the whole genetic makeup under the transcriptional umbrella of HIF-1 alpha to be activated. Now, the question is how this pathway has been triggered. We mentioned inflammatory stress, but what are the dots in terms to understand the mechanistic basis? So which brings this HIF-1 alpha and PFKB3? What's the way to stabilize them? So from the name, HIF-1 alpha, hypoxia-inducible factor 1 alpha, it is obviously implied that hypoxia is the main trigger of this pathway. This pathway is very well established in context of cancer, but the question is whether this is the case in diabetes, and in here in particular in type 1. And again, let me bring again the commonalities between type 1 and type 2 diabetes. Interestingly, I don't know whether we are aware of that or not, but there is shared commonality in terms of perturbation of calcium homeostasis in type 2 diabetes that often comes from misfolded protein stress by IPP, so islet amyloidogenic pancreatic polypeptide. But in context of pro-inflammatory stress, it has been published that also cytokines can lead to perturbation of calcium, intracellular calcium, and mainly mechanism is actually depletion of ER stores while reinforcing higher influx of calcium from outside intracellularly into beta cells. So therefore, there is the shared commonality. On the other side, in type 2 diabetes, we have done a very profound metabolomics and also glucose tracing analysis to find out that actually critical point in stabilization of this pathway is reshaping or a so-called defensive posture that mitochondria, in order to defend itself from calcium waves, attains, and which leads to attenuation of its function. In other words, the TCA cycle. As a consequence of which, we see accumulation of metabolites that we followed through metabolomics analysis, such as succinate and fumarate, that mimic hypoxia on metabolism level and thus stimulate via post-translational mechanism stabilization of HIF-1 alpha. So now we ask the question whether this is the case in context of type 1 diabetes, and we focused on mitochondria for this reason. We have taken here again pancreata from the high quality endpoint collection. For non-diabetics, our pre-diabetic case that we described, published, reported by Dr. Peter Butler, and about, I think, 5 or 6 donors with type 1 diabetes, and measured changes after visualization of mitochondria with TOM20. And we found that mitochondria show decrease in density and also profound fragmented nature. And this was similar in terms of fragmentation, what we observed in response to calcium toxicity in type 2 diabetes. So mitochondria qualify in terms through this change to attenuation that can lead to compensatory stabilization of HIF-1 alpha and PFKB3, similar to what was observed in type 2 diabetes. Now, the question is where all these statements, how they fit when we in vivo manipulate HIF-1 alpha and PFKB3. And I will at this point refer to very preliminary format of our experiments in which we have used streptozotocin as a model that induces injury up to 60% in beta cells, and we have used standard protocol with low dosage over the course of 5 days. But prior that, we established cohorts of mice where we have either genetically knocked out PFKB3 specifically to beta cells, or those which will be presented here in violet, where 9 days post the injury, we administered intraperitoneally established experimental tool inhibitor of PFKB3 for only 4 days. And then we measured metabolic performance of these mice. So we found that there was a profoundly decrease in the excursion of glucose and improvement of glucose tolerance in the groups where either prior the injury, PFKB3 was knocked out, or inhibited 9 days post-injury, and this is quantified under C. As I say, I will be very cautious to make any conclusions here, but they are very similar results to what we have produced and published in context of type 2 diabetes. Now, I would like to turn your attention to inhibition of PFKB3 in context of measurement of consequences to autoimmunity. It is, I don't know whether enough it is reiterated in the literature or it is known to this audience, but absolutely one of the critical steps in creating of immunocompetent effector T cells is basically for their conversion from naive to effector cells. Important is aerobic glycolysis, and in particular both HIF-1 alpha and PFKB3. So that was the premise that recently in 2021 in Frontiers of Immunology, one group tested using the model of adoptive transfer of auto-activated T cells, tested the effect of systemic inhibition of PFKB3 with this, again, experimental tool compound called PFK15. They found when they measured that they have prevented or slowed down the onset of type 1 diabetes. And this was in connection with no gross changes, at least in the body weight, indicating no gross toxicity, which led to differences between the treatment and the vehicle group. But also, we were intrigued to look at what's happening in the islets, on the level of the islets in these mice. And you can compare here the vehicle versus the treatment group, where obviously the consequences of the treatment were that the profound and very diffuse insulitis was restricted to peri-islet stage 1 insulitis, but also there was a reemergence of the beta cells. And interestingly, that was in relation to containment of autoimmunity, as it was evidenced by reduction of autoactive T cells, or what they called exhaustion of these autoactive T cells, in particularly in the spleen. Now, this is very interesting observation. And at this point, I would just like to briefly summarize what we have seen and what I have shown you, that HIF-1 alpha PFKB3 pathway promotes survival of injured beta cells at the expense of their function in context of type 1 diabetes, that HIF-1 alpha PFKB3 immunopositive beta cells account for 15 to 20% in prediabetes and increase up to 2/3 of all beta cells in type 1. And that, of course, these thoughts together with the idea that PFKB3 and HIF-1 alpha also are relevant for the autoimmune response pose the question, is it beneficial to eliminate those HIF-1 alpha PFKB3 immunopositive beta cells? Because obviously these are injured beta cells that can sustain the injury, pro-inflammatory injury, because of upregulation of this pathway. So I would like to propose to you to reflect on a new concept that we are working now, and in which we are inferring certain results at this point of preliminary work in type 1 diabetes from the model of type 2 diabetes. But it's a very interesting concept, and therefore I would like to share it with you. It's basically funded on the activation of very specific beta cell battle, the battle of which selects the fittest, and which is called a cell fitness competition. So the idea of existence of this particular phenomenon in post-developmental tissue in context of disease comes from our observation, and I will apologize for my digression again back to type 2 diabetes, but our observation that we recently published in context of type 2 diabetes, namely when we have knocked out specifically PFKB3 on type 2 diabetogenic stress, which we call humanized mouse model of type 2 diabetes because of bringing together misfolded lipotoxic stress and old age. We have found there was no change in beta cell mass when we inspected the islets from those mice. However, we have seen shift in favor of beta cells relative to alpha cells, and very interesting concomitant phenomenon that described increase in replication of beta cells, at the same time diminishment of damaged or injured cells by misfolding protein stress, but also diminishment of bihormonal cells that occurred concomitantly with increase of replication. This picture is fully reminiscent of a phenomenon that I would like now to share with you, and which basically is a novel concept that we propose as a potential impact for introducing regeneration in post-mitotic tissue. This concept is called cell fitness competition, and basically proposes existence and discrimination between losers and winners in the cellular community. What does it mean is that it proposes that injured, damaged, and old cells are constantly recognized by their neighboring healthy cells, and through extrinsic mechanism, they are expelled from the tissue. Apoptosis is induced in these cells, so that when they open free space, they signal through tangential forces to neighboring healthy cells to reconstitute this free space only quid pro quo, which means with respect and appreciation of physiological constraints of size and space, and how replication ceases or stops as soon as they sense the cell-cell contacts or cell contact inhibition. As soon as this is accomplished, that neighboring healthy cells reconstitute the free space from expelled damaged cells. So fitness is a matrix of wealth in cellular community, and it's based on 3 generic categories: resources, similar to society, protein biosynthesis, energy, the way energy is generated in the cell, and infrastructure, which is mitochondrial form and function. So what we propose and what we want to work, our working model both in type 1 and type 2 diabetes, is the following. We propose that this kind of post-mitotic extrinsic quality control goes on in the endogenous organs such as pancreas, yeast, and pancreatic islets. So the difference is when you have a skin exogenous organ, massive death and massive reconstitution with replication rate. These quite diminished rates of replication and apoptosis go hand in hand when you look, and they reflect very much cell fitness competition. We believe this is active in healthy non-diabetics, but chronic injury changes the makeup and leads basically to retainment of cells which possess the injury. And it has been shown, for example, in Anil Bhushan work, those cells change. They can even change the secretome. They can be all damaged, but it doesn't matter what they belong to, because this is opportunistic concept. And what it matters, what we ask is what makes them to survive and be retained of the tissue, because we believe that they qualify ultimately to contribute to islet failure, and with these 2 onset of diabetes, both type 1 and type 2 diabetes. Now, we think that HIF1 and PFKB3 qualify for a mechanism that conceal their loser status, which it basically interferes with their recognition and expelling from the tissue, leads to their accumulation. So we propose that targeting of this pathway is a rational strategy to initiate their elimination of the tissue and basically rely on intrinsic property of evolutionary conserved principle to initiate functional cell regeneration by enrichment of the tissue with healthy functional cells. So if we are working on that in context of type 1 diabetes, and if this happens to be true, as much as we see that it applies to type 2 diabetes, then interesting here is that we potentially are up to the strategy that can actually cover the 2 features, both features of the pathogenesis of type 1 diabetes, islet failure, as well as T-cell autoimmunity. So with this, I would like very much to thank you for your attention, to thank to all the people who worked on this project, so related between type 2 and type 1 diabetes, my collaborators, the daily source of my wisdom at the Hillblom Islet Research Center, Dr. Peter Butler and Dr. Gerlo, funding, and very happy to take your questions on. Thank you very much.
That was fantastic, really interesting. We've had Peter Thompson as well as Neil Bouchon speaking to us before, and you're kind of bringing a lot of the research angles together in this talk. It was really great to see that kind of put together and highlighted. And I think I'm also going to say I see here that there's a postdoc position available and a research associate position available. So that's amazing. If anyone's hearing this and they would like to reach out to Tudzarova at mednet.ucla.edu, please feel free to do so. And here's a question I'm going to take from the chat. That was a wonderful talk and data. Are you considering the change in immune cells in response to STZ? Unless islet failure is being studied in immunosuppressive mice, STZ may impact immune function and thereby beta and alpha cell ratio. That's the first one. Would you like to take it?
Yes, so we are aware that streptozotocin model is not ideal model of, ideal model of type 1, of human type 1 diabetes, and that pro-inflammatory stress is post-injury. So basically it's a consequence of the massive injury of beta cells. But basically, we will expand on more models than this streptozotocin. And as I say, it's a very preliminary set of data. I cannot comment on how this, definitely the basic mechanism which we rely on is partial inflammation induced after 60% elimination of this beta cell post this alkylating agent injury. So whether this differentially affects beta and alpha cells, we have not yet looked at it, and I don't know whether in the literature that has been pinpointed. But here, most important thing when it comes to articulation of this cell fitness competition is that you have to have residual healthy beta cell mass. So no matter of the rearrangement between beta and alpha cells, if you have residual beta cell mass and within homotypic population, apparently beta cells, if they are injured relative to healthy beta cells, which are juxtaposed, you are in the best position to basically use this to activate this cell fitness competition, we believe, if you deprive injured cells from their survival mode. So ultimately, I find your question is valid, but I cannot say we have not investigated yet the rearrangement between alpha and beta cells as a consequence of streptozotocin. But what it matters is not really that rearrangement. What it matters is preservation of residual beta cell mass in order to articulate cell fitness competition.
That's, excellent answer. I think that was great. And then the answer from Curdy Call was like, that's fair. We've reported insulin resistance in NOD mice too. So reproducing this in other models will be convincing. Couple of claps, interesting and amazing work, people were saying. And I also wanted to ask you, how does your model fit with the prodrome progression? And when we're talking about senescent cells, right? I mean, the sort of the one hit viral or some environmental hit, and then a recovery or remission, if you want to call it, and then another hit, and then a recovery, and those correspond with presentation of autoantibodies, and then the final hit and the final, progression to diagnosis. How might senescent cells, be progressing alongside that model? Have any just hypotheses or data to share?
So far, we are investigating. Ultimately, it's not really important the identity. These senescent cells, I think it's appearance of senescent cell, to my understanding, is relevant only when the first hit is early during development of beta cells, so they replicate. And senescent is a program that is adapted to prevent fixation of genetic lesion in replicating cells. not necessarily, if this happens in adult post-development, not necessarily this is senescent. Because, I mean, to my understanding, replication is important in order to shift the phenotype to senescence. However, in terms of what you say, it is definitely a dynamic process. This self-fitness competition is opportunistic process. So as you can see, so there are a lot of changes that ultimately go and define this relationship between the healthy and less fit, or the cells with lesser fitness. And that can be dynamic, that can be over the course of time, very different. So, but ultimately the trend from what we see is accumulation of these injured cells that change sufficiently to sustain the injury and then accumulate to a proportion that is then sufficient for ultimately onset of hyperglycemia and sincere type 1 diabetes. So no matter, I can imagine, I don't have insight into the particular dynamic of this process, but because that it is highly opportunistic in itself. So basically it's selection of the fittest, which implies that this loser, this injured cell population, can acquire changes dynamically over the course. And with this, what we propose is ultimately conceal this loser status, so that when it is formed in a bulk sufficient to mediate islet failure or beta cell dysfunction, that from the site of the islet contributes to development of type 1 diabetes. So I would not have, for that, we have to follow the kinetic of it more appropriately. It's a very valid question, but I would think from the nature of what I have explained, it is imminent, it's highly opportunistic process that it can be dynamic, and this acquiring of the hits, I might understand as acquiring of ultimate concealment of their recognition by and expelling from the tissue by healthy cells.
Yeah, it's almost like they may, I mean, just sort of very 5,000-foot view. It's almost like perhaps they can clear, these senescent cells, and then the next insult, maybe they can clear less senescent cells and then not at all, or something along those lines.
So along this pressure, which is coming from adversity of the, let's say it's pro-inflammatory, in concurrent pro-inflammatory stress, or other types of stresses, along that this senescent, or if they're senescent or whatever, the damaged injured cells, which we call of lesser fitness or losers, are exerted to the same, just in a shorter time, evolutionary pressure to change. And this change is dynamic because the process is opportunistic. And the point is that what is dangerous is when they cannot be any more actively expelled from the tissue. So if they, in the beginning, to a certain extent, they are eliminated by the, by this active self-fitness competition, that over the course of chronicity of the stress might be lost and linked. So to, for example, changing the way they generate the energy, which is suppressing oxidative stress and masking their existence in their beta cell population.
I guess that's so interesting. I wanted, last question from me at least is, how does the presence of senescence in the islets kind of correspond, or even exist under the umbrella of the HLA differences and the HLA backdrop of type 1 diabetes?
Yeah, so from what I read, that basically this HLA risk alleles, right, 10% only of bearers of these risk alleles ultimately develop type 1 diabetes, but that percentage is even declining, which implicates, other gene environmental interactions that may play a huge role. It will be interesting whether this is articulating the status of recognition and elimination of senescent and injured cells. But I can tell you what the literature is indicating. It might be, it might be not, but much more focus so far is number one, it's a prototoxicity, and number 2 is oxidative stress. So the reason why this HIF-1 alpha PFKB3 is chosen to conceal during chronic stress, to conceal the loser status, is because it's one of the most powerful ways by switching to oxygen-independent way of generation of energy to suppress mitochondrial byproduct, mitochondrial OXPHOS byproduct, which is reactive oxygen species, and with which these loser cells actually can conceal the status. So they are more kind of, related so far in the literature and in genetic lower organisms, which are more appropriate for mechanistic studies, such as Drosophila. It has been shown that ER and UPR stress ultimately through readout of proteotoxicity, protein aggregates, and oxidative stress are defining this status. So I don't know how much.
That's fantastic. That's really interesting. No, that is really, really, that's a very interesting point. And I think, I would encourage anyone to really sort of connect with you, to dive into what it is you're doing and what is available for a research associate or postdoc, because this work is really quite cutting edge. Peter Butler, yourself, Peter Thompson up in Canada, and Anil Bhushan are really kind of pioneers in this realm. I believe Kirtikal was on here as well. And this is a really novel approach and a really exciting place to be right now. So thank you again for sharing this innovative research, and we cannot wait to see what comes from your lab and your collaborations next. Thank you again.
Thank you so much, Monica. I appreciate, and everyone else, and thank you for making this visible work with every word and every kind of this support further encouraged. Thank you so much.
Absolutely. Have a great rest of your day. Bye-bye.
Thank you. Bye-bye.